There is a small coastal town in Japan where it is completely normal to see a 93-year-old woman walking to the market alone, carrying her own groceries, preparing her own meals, and going to bed at night without taking a single painkiller. Not because she is exceptional. Not because she won some genetic lottery. Not because she lives near some mineral-rich spring or follows some ancient spiritual discipline. But because what she does every single day, the specific foods she eats, the way she moves through her environment, the time she wakes up, the time she eats, the time she sleeps, has been creating a very specific biological environment inside her body for the past 7 decades. And that environment, maintained consistently and without interruption across a lifetime, does not allow chronic pain to take root the way it does in populations that live differently. This is not a rare case. In Okinawa, in the rural regions of Nagano, in the fishing villages along the coast of the Sea of Japan, you find the same pattern repeated. Men and women in their late 80s and 90s who remain functionally independent, cognitively sharp, and largely free from the chronic musculoskeletal conditions that are considered almost inevitable in Western medicine once a patient passes 75. They are not free from all illness. They are not immortal. But the specific kind of deterioration, the joint pain, the inflammatory disease, the early dependence on pharmaceutical management, that is the baseline reality for most aging adults in the United States, is strikingly, measurably less prevalent among them. Meanwhile, in the United States, more than 70% of adults over the age of 65 live with chronic joint pain severe enough to limit their daily activities in some meaningful way. More than half of adults over 75 take at least 5 prescription medications daily. Millions depend on prescription anti-inflammatories just to get through the morning routine without significant discomfort. Millions more require some form of assisted care by their mid-70s, not because of acute illness or injury, but because the gradual accumulation of inflammatory and degenerative damage over decades has reduced their functional capacity below the threshold of independent living. The gap between these two realities is enormous, and it is not explained by genetics. This is one of the most important points I want you to understand before we go any further. When researchers study Japanese Americans, people of full Japanese ancestry who were born or raised in the United States and adopted Western dietary and lifestyle patterns, they find that those individuals develop essentially the same chronic disease profiles as any other American population. Their rates of cardiovascular disease, type 2 diabetes, osteoarthritis, and cognitive decline match the American average far more closely than they match the Japanese average. The biology is identical. The ancestry is identical. What changes the outcome entirely is the daily input, the food, the movement, the schedule, the environment the body lives in day after day, year after year. My name is Dr. Kenji, and I have spent over 40 years as a physician working in the fields of longevity and preventive medicine, with a specific clinical focus on elderly Japanese populations. I have followed patients into their 80s, their 90s, and in several remarkable cases, past 100 years of age. I have documented their habits, analyzed their dietary patterns, reviewed their laboratory values, and compared their functional outcomes against international data. What I am sharing with you in this video is not a theory assembled from popular health books. It is the product of decades of direct clinical observation, cross-referenced against the most rigorous longevity research available in the medical literature. I want to ask you to stay with me through this entire video. The reason is structural. Each section of what I am going to explain builds on the one before it. The individual habits I will describe are meaningful on their own, but their combined effect is far greater than any single element. The biology of aging and inflammation does not operate through isolated variables. It operates through systems. And to understand why Japanese elderly remain pain-free and functional at ages when most Western adults have already surrendered significant portions of their independence, you need to understand the system, not just the parts. Everything I am about to explain is practical. None of it requires you to move to Japan. None of it requires expensive supplements, specialized equipment, or dramatic changes implemented overnight. What it requires is understanding, a clear, mechanistic understanding of what is happening inside the body and why these specific habits produce these specific outcomes. Once you understand the mechanism, the habit makes sense. And habits that make sense are the ones that actually get maintained. Let us begin where the Japanese elder begins, at the first meal of the day. If you were to sit at the breakfast table of a Japanese man or woman in their late 80s or early 90s and observe what they eat within the first hour of waking, you would notice immediately that what is in front of them bears almost no resemblance to what most American adults consume at that same hour. There is no cereal with pasteurized milk. There is no toast with butter and jam. There is no glass of orange juice, no flavored yogurt, no processed breakfast bar eaten in the car. What you find instead is a small ceramic bowl of miso soup, typically containing cubes of soft tofu, a small amount of seaweed, perhaps a few slices of green onion. Beside it, a portion of natto, fermented soybeans with a sticky texture and a sharp, fermented smell. A small piece of grilled fish, often mackerel or salmon. A modest bowl of short-grain white rice or sometimes barley. And beside all of it, a cup of green tea, prepared simply, consumed slowly. This meal has not been designed by a nutritionist. It has not been assembled based on the latest dietary guidelines from a health authority. It is simply what has been eaten in that household, and in households like it across Japan for generations. And yet, if you were to reverse engineer this meal from a purely clinical standpoint, if you were to ask what combination of foods would best protect a body over 60 from the specific biological deteriorations that accompany aging, you would arrive at something remarkably close to what is sitting on that table. To understand why, you need to understand what happens to the gut after the age of 50. This is a change that receives far less attention in Western preventive medicine than it deserves, and it is one of the most consequential biological shifts that occurs as the human body ages. The lining of the small intestine is composed of a single layer of epithelial cells, held together at their edges by structures called tight junctions, protein complexes that seal the spaces between cells and form a selective barrier between the contents of the intestinal canal and the bloodstream. This barrier allows nutrients to pass through in controlled quantities while preventing bacteria, undigested food particles, and microbial waste products from entering circulation. When this barrier is intact and functioning correctly, it is one of the body's most powerful anti-inflammatory defenses. As we age, the proteins that form these tight junctions, occludin, claudin, zonulin-regulated complexes, begin to degrade. The integrity of the barrier diminishes. The gaps between intestinal cells widen, and substances that were previously excluded from the bloodstream begin to pass through in increasing quantities. Bacterial lipopolysaccharides, fragments of the outer membrane of gram-negative bacteria that are normally confined to the intestinal canal, enter circulation and trigger a powerful immune response. The immune system identifies these fragments as a threat and responds with inflammation. This inflammatory response does not stay localized. It becomes systemic. It circulates. It reaches the synovial lining of joints, producing pain and stiffness. It reaches the endothelium of arteries, promoting plaque formation. It crosses the blood-brain barrier and contributes to neuroinflammation associated with cognitive decline. This condition, increased intestinal permeability combined with the downstream systemic inflammation it produces, is increasingly recognized in the medical literature as a root driver of many of the chronic diseases that disproportionately affect aging Western populations. And it is directly and powerfully addressed by what the Japanese elder eats for breakfast every single morning. Miso, which is produced through the fermentation of soybeans with specific strains of bacteria and fungi, primarily Aspergillus oryzae, contains live microbial cultures that contribute to the diversity and resilience of the gut microbiome. A diverse microbiome is not simply a health trend phrase. It is a clinical description of a gut environment that is functionally capable of maintaining the intestinal barrier, outcompeting pathogenic bacteria, and producing the metabolic byproducts, specifically short-chain fatty acids such as butyrate, propionate, and acetate, that serve as the primary fuel source for the epithelial cells of the intestinal lining. Butyrate, in particular, has been shown in multiple clinical studies to directly upregulate the expression of tight junction proteins, reinforcing the intestinal barrier at the cellular level. When the epithelial cells of the intestinal lining have adequate butyrate supply, produced by the fermentation of dietary fiber by beneficial gut bacteria, they maintain the structural integrity that prevents inflammatory molecules from entering the bloodstream. When butyrate production declines because the microbiome has been disrupted or the dietary fiber that feeds it is absent, barrier integrity declines with it. Natto contributes its own distinct bacterial populations, primarily Bacillus subtilis var. Natto, that further diversify the gut environment and have been shown to produce enzymes that support the breakdown of potentially harmful compounds. The tofu in the miso soup provides isoflavones, plant-derived compounds with demonstrated anti-inflammatory activity, as well as a source of easily digestible protein that does not place a high digestive burden on the gastrointestinal system in the morning hours. The fish that accompanies this breakfast, typically a small portion of mackerel, sardine, or salmon, provides omega-3 fatty acids in the form of EPA and DHA. These are the precursors to a class of molecules called resolvins and protectins, which are among the most potent anti-inflammatory compounds the body can produce endogenously. EPA and DHA from marine sources are incorporated directly into cell membranes throughout the body, including the membranes of immune cells. When these membranes contain high concentrations of omega-3 fatty acids, the immune response to inflammatory stimuli is measurably more controlled and more rapidly resolved. And the green tea that accompanies all of this, consumed not as a stimulant but as a quiet daily ritual, begins delivering its anti-inflammatory compounds to the bloodstream before the first hour of the day has ended. What this breakfast constructs from the very first meal is a biological environment oriented toward barrier protection, microbiome support, and inflammation suppression. Every element has a mechanism. Every mechanism addresses a specific biological vulnerability that increases with age. The Japanese elder eating this meal every morning for the past 50 years has been performing a daily act of biological maintenance so consistent and so precisely targeted that it has, over time, produced outcomes that look, to someone who does not understand the mechanism, like remarkable good fortune. Now I want to isolate one specific component of that breakfast and give it the detailed explanation it deserves. Because among all the foods in the traditional Japanese diet that contribute to longevity, natto occupies a position of unique clinical significance. It is not simply a fermented food with gut health benefits. It is one of the most concentrated natural sources of a specific compound that addresses two of the most dangerous biological processes that accelerate after the age of 60: bone mineral loss and arterial calcification. The compound is vitamin K2, and the specific form found in natto, called -7 Malawian quachas, or menaquinone 7, is the most bioavailable and longest-acting form of K2 that exists in any natural food source. To understand why this matters, you need to understand what happens to calcium metabolism in the aging body. Most people are aware that calcium is important for bone health, and most people know that vitamin D is necessary for calcium absorption from the digestive tract. What is far less widely understood is that absorbing calcium is only half of the problem. The other half, the half that determines whether aging adults develop strong bones or brittle ones, clear arteries or calcified ones, is where the absorbed calcium goes once it is in the bloodstream. In a young, healthy body with adequate vitamin K2 levels, calcium is efficiently directed to the skeletal system, where it is incorporated into the bone mineral matrix through a process managed by a protein called osteocalcin. Osteocalcin is produced by osteoblasts, the cells responsible for building new bone tissue, but it can only perform its calcium binding function when it has been activated by vitamin K2 through a process called carboxylation. In the presence of sufficient K2, osteocalcin is carboxylate and functional. It binds calcium ions and anchors them into the crystalline structure of bone. In the absence of K2, osteocalcin remains uncarboxylated and inactive, cannot bind calcium, and the calcium that was absorbed from food, circulating in the bloodstream, has nowhere controlled to go. At the same time, a second K2 dependent protein called matrix GLA protein, MGP, is responsible for preventing calcium from depositing in soft tissue, particularly in the inner walls of arteries. MGP must also be carboxylate by K2 to function. When K2 is sufficient, active MGP inhibits the crystallization of calcium within arterial tissue, keeping arterial walls flexible and clear. When K2 is deficient, MGP is inactive and calcium crystallizes within arterial walls, a process called vascular calcification, which stiffens the arteries, raises blood pressure, and dramatically increases the risk of cardiovascular events. This means that vitamin K2 deficiency creates a situation in which calcium simultaneously fails to reach the bones and accumulates in the arterial walls. Osteoporosis and arterial disease, two of the most common and most dangerous conditions in aging Western populations, are both accelerated by the same nutritional deficiency. And natto, consumed daily in even modest portions, provides more -7 Malawian kwachas than any other food source on the planet. Studies conducted specifically in Japanese populations have found that regions with consistently high natto consumption show significantly lower rates of hip fracture, the most clinically consequential consequence of osteoporosis in the elderly, as well as lower rates of arterial calcification and cardiovascular mortality, compared to regions of Japan where natto is not traditionally consumed. This is a natural dietary experiment of considerable clinical value, because it isolates the variable across a population with otherwise similar dietary and lifestyle patterns. The practical implication for anyone reading this outside Japan is straightforward. Natto is available in frozen form in many Asian grocery stores. The flavor is sharp and the texture is unusual for those unfamiliar with it, but the dose required to produce meaningful -7 Malawian kwachas levels is not large, approximately 80 to 100 grams per day, which is roughly one standard serving. Consumed consistently, not occasionally, this single food addresses two of the most serious threats to the structural integrity of the aging body. The third habit I want to address is one that, on the surface, appears almost too simple to be the subject of serious clinical discussion. It is walking, not structured exercise, not a prescribed fitness regimen. Simply walking, the kind of low-intensity, daily, purposeful movement that Japanese elderly integrate into the fabric of their lives so completely that it never occurs to them to think of it as exercise at all. But the clinical significance of this distinction between walking as a structured exercise event and walking as a continuous feature of daily life is profound. And to understand it, you need to understand the biology of joint cartilage maintenance, because this is one of the mechanisms most directly responsible for the absence of chronic joint pain in elderly Japanese populations. Cartilage, the smooth, cushioning tissue that covers the ends of bones within every joint, has a biological property that makes it uniquely dependent on mechanical stimulation for its survival. Unlike most other tissues in the body, cartilage has no direct blood supply. There are no blood vessels running through it. It receives all of its nutrients, oxygen, glucose, amino acids, growth factors, through a process of diffusion driven by mechanical compression. When the joint bears weight and the cartilage is compressed, synovial fluid is pushed out of the cartilage matrix into the joint space. When the weight is removed and the cartilage decompresses, fresh synovial fluid is drawn back in, bringing nutrients with it. This cycle of compression and decompression is the only mechanism through which cartilage receives its nutritional supply. This means that a joint which is not regularly moved and loaded is a joint whose cartilage is being chronically undernourished. Waste products accumulate within the cartilage matrix. Inflammatory cytokines, particularly interleukin-1 beta and tumor necrosis factor alpha, concentrate within the joint space without the diluting and clearing effect of synovial fluid circulation. Chondrocyte, the cells responsible for maintaining and repairing cartilage, begin to shift from a repair mode to an inflammatory mode. The collagen framework of the cartilage begins to break down faster than it can be rebuilt. The cartilage thins. The joint space narrows. Pain develops. And that pain, in the absence of movement, creates a vicious cycle in which the person moves less, the cartilage deteriorates further, and the pain increases. This is the trajectory that most aging Western adults follow. Not because of any inevitable biological law, but because the structure of Western daily life, desk work, car transportation, sedentary leisure, scheduled but infrequent exercise sessions separated by long periods of complete inactivity, does not provide joints with the continuous, low-level mechanical stimulation they require to maintain cartilage nutrition. Japanese elderly, particularly those living in traditional residential environments, walk continuously throughout the day. They walk to purchase food. They walk to visit neighbors. They walk to community centers, to local shrines, to the houses of family members. In many traditional Japanese neighborhoods, the physical layout of the environment requires regular stair climbing, navigation of uneven terrain, and sustained upright movement throughout the day. The joints are not subjected to extended periods of mechanical inactivity. The cartilage compression-decompression cycle is never interrupted for hours at a time. Beyond cartilage, there is another critical benefit of this pattern of continuous daily ambulation that is particularly relevant after the age of 50. Walking at low to moderate intensity is one of the most effective stimuli for lymphatic circulation. Unlike the cardiovascular system, the lymphatic system has no pump, no heart to drive its fluid through the body. Lymphatic flow depends entirely on the mechanical compression of lymphatic vessels by surrounding muscle tissue during movement. When a person walks, the rhythmic contraction of leg and hip muscles compresses the lymphatic vessels in those regions, driving lymphatic fluid upward through the system. This carries inflammatory mediators, cellular waste products, and excess interstitial fluid away from peripheral tissues, including joint tissue, and toward the lymph nodes where they can be processed and cleared. In a person who is largely sedentary, this clearing process is sluggish. Inflammatory compounds accumulate in joint tissue not only because they are being produced, they accumulate because they are not being efficiently removed. Daily walking, distributed across the natural rhythm of life rather than compressed into a single exercise session, maintains this clearing mechanism continuously. The result, over years and decades, is a joint environment that is consistently less inflamed and more nutritionally replete than what is found in sedentary populations of the same age. The fourth element of this system addresses something that most people over 50 are already aware of, the progressive loss of muscle mass, but from an angle that most nutritional advice, even well-intentioned advice, consistently gets wrong. The critical variable is not simply how much protein you eat. It is when you eat it and how that timing interacts with the biology of muscle protein synthesis in the aging body. Sarcopenia, the clinical term for the age-related loss of skeletal muscle mass and function, begins in most people during their 40s and accelerates significantly after 60. By the age of 70, an individual who has not actively counteracted this process through diet and movement may have lost 25 to 30% of the muscle mass they had at their peak. By 80, that figure can approach 40% in sedentary individuals. This matters far beyond appearance or strength. Skeletal muscle is the primary site of insulin-stimulated glucose uptake in the body. When muscle mass declines, the body's capacity to manage blood glucose diminishes, insulin resistance increases, and the risk of type 2 diabetes rises sharply. Muscle is also the primary mechanical stabilizer of every joint in the body. As muscle mass declines, the mechanical load on cartilage and connective tissue increases disproportionately. And muscle is the primary defense against the falls that, in people over 75, represent one of the leading causes of serious injury and loss of independence. The standard advice regarding protein for older adults focuses almost exclusively on total daily intake. Consume enough grams of protein per day. This advice is not wrong, but it is incomplete in a way that makes it far less effective than it could be. Here is what the research actually shows. Muscle protein synthesis, the process through which the body repairs existing muscle tissue and constructs new muscle protein, is not simply a function of how much amino acid substrate is available over the course of a day. It is triggered by the presence of amino acids in the bloodstream above a specific threshold at a given point in time. When amino acid levels in the blood rise above this threshold, which happens when you consume a meal containing adequate protein, a molecular signaling cascade is initiated, primarily through a pathway called mTOR, that activates the cellular machinery for protein synthesis. This synthesis window lasts for approximately 3 to 5 hours following the meal. Then the signal diminishes, regardless of how much protein was consumed at that meal, until amino acids are again elevated by another protein-containing meal. This means that consuming a very large amount of protein at one meal, say a large steak at dinner, does not produce a proportionally larger or longer anabolic response than consuming a moderate amount. The mTOR pathway saturates. Excess amino acids beyond what the synthesis response can use are simply oxidized or converted to glucose. And if the other meals of that day contained minimal protein, the body spent the majority of its waking hours in a state where the muscle protein synthesis signal was absent, which means that any breakdown of existing muscle tissue that occurred during those hours was not being offset by new synthesis. This is precisely the pattern that most Western older adults follow. Protein intake is concentrated at dinner, with relatively low protein at breakfast and lunch. The result is that for 12 to 14 hours of the day, including all of breakfast and lunch, the anabolic signal is absent, and the body is in a net catabolic state relative to muscle tissue. Japanese elderly distribute protein intake across all 3 meals in a way that ensures the mTOR signaling pathway is activated multiple times throughout the day. Breakfast includes fish, tofu, or egg, all high biological value protein sources. Lunch includes another protein source, frequently fish again, or a combination of legumes and a small amount of meat or seafood. Dinner is similarly moderate and protein-containing. The result is that the anabolic signal is activated in the morning, again at midday, and again in the evening. The body spends a far smaller proportion of its waking hours in a catabolic state relative to muscle tissue. The specific protein sources matter as well. Fish provides a complete amino acid profile with particularly high concentrations of leucine, the specific branch-chain amino acid that has been identified in clinical research as the primary trigger for mTOR activation. Leucine has a threshold effect. Below approximately 2 to 3 grams per meal, the mTOR pathway is not robustly activated. Above that threshold, activation occurs reliably. A typical serving of fish or eggs provides leucine in quantities sufficient to cross this threshold. Tofu and other soy-based protein sources, when consumed in sufficient portions, also provide the necessary leucine content along with isoflavones that have demonstrated specific muscle-preserving effects in older adults in clinical trials. The combined effect of distributed protein intake from high-quality sources, in a body that is also receiving the mechanical stimulus of daily walking, is a rate of sarcopenia that is dramatically slower than what is observed in Western populations of the same age. This is not speculation. It is consistent with the clinical and anthropological data on Japanese elderly populations across multiple longitudinal studies. Green tea is the next element I want to discuss, not as a comfortable warm beverage with vague health associations, but as a pharmacologically active substance consumed in a pattern that produces sustained, measurable anti-inflammatory effects throughout the day. Understanding green tea clinically requires understanding its primary active compound and the cellular mechanism through which that compound operates. Epigallocatechin gallate, EGCG, is the most abundant and most studied polyphenol in green tea. It belongs to a class of compounds called catechins, and its biological activity is remarkable in its specificity. EGCG has been shown in multiple cell culture, animal, and human clinical studies to inhibit the activation of NF-κB nuclear factor kappa light chain enhancer of activated B cells, which functions as a master regulatory switch for the production of pro-inflammatory cytokines throughout the body. When NF-κB is activated, as it is by bacterial fragments entering the bloodstream through a permeable intestinal barrier, by oxidized lipids from processed seed oils, by advanced glycation end products from high-sugar diets, and by a range of other inflammatory triggers that are common in Western dietary and lifestyle patterns. It initiates the transcription of genes that produce interleukin-1 beta, interleukin-6, tumor necrosis factor alpha, and a cascade of other pro-inflammatory signaling molecules. These cytokines, when chronically elevated, drive the inflammation that erodes cartilage, stiffens arteries, disrupts insulin signaling, and, in the brain, promotes the neuroinflammation associated with cognitive decline and dementia. EGCG interferes directly with the NF-κB activation pathway, reducing the transcription of these inflammatory genes. It also inhibits a class of enzymes called matrix metalloproteinases, specifically MMP1, MMP3, and MMP13, which are the primary enzymatic drivers of cartilage matrix degradation. These enzymes break down collagen type 2, the structural protein that gives cartilage its load-bearing properties. In joint tissue exposed to chronic low-grade inflammation, matrix metalloproteinase activity is chronically elevated, and cartilage degrades faster than it can be repaired. EGCG suppresses this enzymatic activity directly within joint tissue. After the age of 50, the baseline level of circulating pro-inflammatory cytokines rises in essentially all adults, a phenomenon that researchers have termed inflammaging. This is not a disease state in the conventional diagnostic sense, but it is the biological background against which virtually every degenerative process of aging occurs. Cartilage erosion, arterial stiffening, insulin resistance, and the early stages of cognitive decline all proceed more rapidly in a high-inflammaging environment. Anything that consistently and reliably reduces this baseline inflammatory tone has correspondingly protective effects across all of these systems simultaneously. This is precisely what consistent green tea consumption does when consumed in the pattern observed in elderly Japanese populations. Not one large cup in the morning, not an occasional cup when the mood arises. 3 to 5 small cups distributed throughout the day, from morning through the afternoon, maintaining a relatively stable plasma concentration of EGCG across waking hours. This sustained plasma level is what produces continuous NF-κB suppression, continuous matrix metalloproteinase inhibition, and the steady anti-inflammatory background that, over decades, meaningfully differentiates the joint and cardiovascular health of populations with high green tea intake from those without it. The practical implementation requires no complexity. High-quality loose-leaf green tea, sencha, gyokuro, or bancha, steeped in water that is hot but not boiling, consumed plain without added sugar, multiple times across the day. The temperature of consumption and the specific variety matter less than the frequency and the absence of sugar, which would introduce an inflammatory stimulus through the same beverage that is meant to reduce inflammation. Now I want to turn to something that requires a different kind of attention, not the addition of a protective habit, but the absence of a destructive one. Because one of the most powerful variables in the longevity of elderly Japanese populations is not found in what they eat. It is found in what they have never eaten across decades, in the quantities that most Western adults consume without a second thought. The traditional Japanese diet contains virtually no processed seed oils. This is not a minor dietary detail. It is a fundamental structural difference with enormous biological consequences. Processed seed oils, refined corn oil, refined soybean oil, refined sunflower oil, refined canola oil, and similar products are produced by industrial processes that expose polyunsaturated fatty acids to high heat, chemical solvents, and bleaching agents. The resulting oils are extraordinarily high in omega-6 polyunsaturated fatty acids, particularly linoleic acid. These fatty acids are inherently chemically unstable. Their multiple double bonds make them highly susceptible to oxidation when exposed to oxygen, light, and especially heat. When these oils are used in cooking, heated in a pan, used in frying, incorporated into processed foods that are then baked at high temperatures, a significant proportion of the polyunsaturated fatty acids undergo oxidation, producing reactive compounds called lipid peroxides for hydroxynonenal and malondialdehyde, among others. When these oxidized lipid compounds are consumed, they do not simply pass through the body harmlessly. They are absorbed through the intestinal wall and incorporated into the circulation. They react with proteins and DNA within cells, producing what is collectively termed oxidative stress. The mitochondria, the organelles within every cell that produce the ATP that powers all cellular function, are particularly vulnerable to this oxidative damage. Mitochondrial membranes are rich in polyunsaturated fatty acids, and when those fatty acids are damaged by reactive oxidized compounds, mitochondrial function degrades. Energy production becomes less efficient. Cells generate more free radicals as byproducts of impaired mitochondrial function. Cellular aging accelerates. This mechanism operates across all tissue types. In cartilage cells, chondrocyte mitochondrial dysfunction impairs the energy-intensive process of cartilage matrix repair. In arterial endothelial cells, oxidative stress triggers the inflammatory cascade that initiates atherosclerotic plaque formation. In neurons, mitochondrial dysfunction is one of the earliest and most consistent findings in the tissue of patients with Alzheimer's disease and other neurodegenerative conditions. In immune cells, oxidative stress dysregulates the inflammatory response, making it both more intense and more difficult to resolve. Japanese traditional cooking uses sesame oil, which, though it contains some polyunsaturated fatty acids, is predominantly used in small amounts and at relatively low temperatures, and contains natural antioxidants including sesamol and sesamin that protect against oxidation. The primary fat sources in the traditional Japanese diet are fish, which provides omega-3 fatty acids with anti-inflammatory rather than pro-inflammatory effects, and the small amounts of fat naturally present in whole foods. The cumulative oxidative burden per meal is dramatically lower than in a diet cooked with refined industrial seed oils. High fructose products represent the second major absence. Fructose, unlike glucose, is metabolized almost exclusively in the liver through a pathway that does not require insulin and is not regulated by the feedback mechanisms that normally govern glucose metabolism. When fructose is consumed in large quantities, as it is in any diet that includes significant amounts of high fructose corn syrup, sucrose-sweetened beverages, or heavily sweetened processed foods, the liver is overwhelmed. Excess fructose is converted to uric acid, which is then released into the bloodstream and deposited in joint spaces, where it crystallizes and triggers the intense inflammatory response of gout. Even at levels below those that produce overt gout, elevated uric acid is associated with joint inflammation, endothelial dysfunction, and impaired kidney function. Simultaneously, excess fructose in the liver is converted to triglycerides through de novo lipogenesis. These triglycerides are packaged into VLDL particles and released into circulation, contributing to the dyslipidemia, specifically elevated triglycerides and reduced HDL, that drives arterial disease. Fructose also directly impairs the function of leptin, the hormone produced by fat cells that signals satiety to the brain. When leptin signaling is disrupted by chronic fructose consumption, the brain's perception of fullness is blunted, promoting overconsumption and the metabolic consequences of chronic caloric excess. The traditional diet of Japanese elderly contains fructose only in the forms naturally present in small quantities of whole fruit, consumed occasionally and in modest portions. The liver is not subjected to the daily high-volume fructose load that is effectively unavoidable for most adults in the United States whose diet includes any significant quantity of packaged, processed, or restaurant food. Ultra-processed food products, which are defined not only by their ingredient profiles but by their industrial formulation, which typically includes emulsifiers, artificial flavor compounds, texture modifiers, and preservatives, are absent from the traditional Japanese elderly diet for the simple reason that these products did not exist within that food culture, and they have been largely resisted or ignored by older generations who formed their dietary habits before industrial food became dominant. The emulsifiers present in many ultra-processed foods, including polysorbate 80 and carboxymethylcellulose, have been shown in animal studies and early human research to disrupt the mucus layer that lines the intestinal epithelium, degrading the physical barrier between the gut microbiome and the intestinal wall. This degradation of the mucus layer increases the contact between bacterial communities and intestinal epithelial cells, triggering inflammatory responses and contributing to the increased intestinal permeability already discussed. The cumulative effect of 70 or 80 years of eating in the way that Japanese elderly eat, not only adding beneficial foods and habits but also, crucially, never loading the body with the daily inflammatory, oxidative, and metabolic burden that is embedded in the Western dietary pattern, produces a body that, at 90 years of age, has simply never had to manage the degree of chronic damage that most Western adults begin accumulating in their 30s. The eighth and final specific habit I want to address is sleep, but not simply the number of hours. What distinguishes the sleep of Japanese elderly from that of Western adults of the same age is not primarily duration. It is regularity, architecture, and the environmental conditions under which it occurs. Every cell in the human body operates on a circadian rhythm, an internal biochemical clock with a cycle of approximately 24 hours, synchronized to the external environment primarily by light and secondarily by the timing of meals and physical activity. The master circadian clock resides in a region of the hypothalamus called the suprachiasmatic nucleus. It receives light input directly from the retina through a dedicated neural pathway and uses this information to synchronize the peripheral circadian clocks operating in every organ system throughout the body, including immune cells, joint tissue, the cardiovascular system, the gastrointestinal tract, and the brain. When this synchronization is maintained, when sleep and wake times are consistent across 7 days a week, when light exposure follows a natural pattern, when meals occur at predictable times, when physical activity is distributed across daylight hours, the body's physiological processes unfold in their optimal temporal sequence. Cortisol, the body's primary endogenous anti-inflammatory hormone, peaks in the early morning hours, providing a natural daily suppression of inflammatory activity. This cortisol peak also serves as a powerful circadian timing signal, reinforcing the synchronization of peripheral clocks throughout the body. As cortisol declines through the day, the immune system becomes more active, not in a pathological sense, but in the sense of performing its maintenance functions, identifying and clearing damaged cells, monitoring for nascent abnormalities, repairing tissue microdamage. During the deepest phases of slow-wave sleep, the pituitary gland releases growth hormone in concentrated pulses. Growth hormone drives the synthesis of collagen, the structural protein that forms the matrix of joint cartilage, tendons, ligaments, and connective tissue throughout the body. It also stimulates the uptake of amino acids into muscle tissue and promotes the repair of microdamage accumulated during the day's physical activity. This nighttime repair window is not a passive process. It is an active, energy-intensive biological maintenance cycle that is as important to the long-term structural integrity of the body as any of the daytime habits we have discussed. And it depends on the integrity of the circadian system to function correctly. When sleep schedules are irregular, when a person sleeps at different times on different nights, stays awake late some nights and retires early others, uses bright artificial light in the hours before sleep, or eats a large meal within 2 to 3 hours of bedtime, the circadian synchronization of peripheral organ clocks is disrupted. The cortisol rhythm flattens or shifts out of phase. Growth hormone secretion during sleep is reduced in both amplitude and duration. The immune system's maintenance activity during sleep is compressed and less thorough. Inflammatory cytokines that should decline overnight remain elevated into the following morning, carrying forward an inflammatory burden that accumulates over time. In adults over 60, circadian disruption has additional consequences that are specific to the aging system. The suprachiasmatic nucleus itself shows evidence of functional decline with age. Its oscillation amplitude decreases, its responsiveness to light input diminishes, and its capacity to maintain tight peripheral synchronization weakens. This means that older adults are inherently more vulnerable to circadian disruption than younger ones. Small inconsistencies in schedule that a 35-year-old body can compensate for without measurable consequence may, in a 75-year-old, produce detectable increases in inflammatory markers and measurable impairment of nighttime tissue repair. Japanese elderly, as a cultural pattern, maintain a remarkable degree of schedule regularity. They wake at the same time every morning, typically early, aligned with sunrise. They eat at the same times. They are physically active during daylight hours and transition to low-stimulation activities in the evening. Evening environments involve modest lighting, quiet conversation, and no consumption of food after the early evening meal has been fully digested. Sleep begins at a consistent hour, in a dark, quiet environment. This is not described as a health practice by the people who live it. It is simply the rhythm of daily life as it has existed in their community for generations. But the biological consequences of maintaining that rhythm without interruption across 70 or 80 years of adult life are profound. The nighttime repair cycle is intact. Collagen synthesis continues efficiently into advanced age. Muscle repair is completed nightly. The immune system conducts its maintenance operations in full. And the inflammatory burden that accumulates from the previous day's physical activity, metabolic function, and environmental exposure is consistently and thoroughly cleared before the next day begins. What I have described across the course of this video are 8 distinct, specific, observable habits. A morning meal that repairs the gut barrier from the first hour of the day. A fermented food that directs calcium to bones and protects arteries. A pattern of daily movement that maintains joint cartilage through continuous mechanical stimulation. A protein distribution strategy that keeps muscle protein synthesis active throughout the day. A beverage consumed multiple times daily that suppresses the master switch of inflammatory gene expression. A dietary framework defined as much by what is absent as by what is present. And a sleep architecture maintained with a regularity that preserves the body's nighttime repair capacity into advanced age. But these are not 8 independent strategies. They are 8 expressions of a single underlying biological principle. Every one of them, through its own specific mechanism, contributes to the same outcome: a reduction in the baseline level of systemic inflammation that the body is required to manage on a daily basis. Inflammation is not inherently destructive. It is the body's primary repair mechanism, and in acute form, the localized redness, heat, and swelling that follows an injury or infection. It is essential for survival. What is destructive is the chronic, low-grade, system-wide inflammatory state that develops when the body is subjected, day after day and year after year, to a dietary pattern that continuously produces inflammatory signals, a movement pattern that allows inflammatory compounds to accumulate in joint tissue without being cleared, a protein intake pattern that allows muscle to slowly atrophy, a beverage environment dominated by sugar rather than polyphenol, a food environment saturated with oxidative compounds, and a sleep pattern that disrupts the nightly inflammatory clearance cycle. Japanese elderly who remain functional and pain-free into their 90s are not living in a biological reality separate from the rest of aging humanity. They are living in a biological reality that their daily habits have constructed and maintained across decades. Their joints are not miraculous. Their arteries are not immune to the passage of time. Their brains are not protected by some genetic advantage unavailable to others. What they have is an environment, a daily biological environment created by what they eat, how they move, and how they sleep, that consistently reduces the inflammatory burden their bodies carry. The human body, at any age, is responsive to its environment. It is not a static machine that deteriorates on a fixed schedule. It is a dynamic system that continuously adapts to the signals it receives. Feed it fermented foods that repair the gut barrier, and the gut becomes less permeable. Move it consistently throughout the day, and the cartilage stays nourished. Give it distributed protein across meals, and the muscle synthesis signal remains active. Provide it with EGCG across the day, and the master inflammatory switch stays suppressed. Remove the oxidative and fructose burden, and the liver and mitochondria function more efficiently. Maintain a consistent circadian schedule, and the nighttime repair cycle runs completely. None of these adaptations are dramatic or rapid in isolation. They are cumulative. They are the product of daily repetition across years and decades. The Japanese elder who at 92 still walks to the market, still cooks her own food, and still wakes each morning without reaching for a painkiller did not arrive at that outcome through luck. She arrived there through the compounding effect of 70 years of daily signals that told her body, consistently and without contradiction, to reduce inflammation, repair tissue, maintain structure and function. That is the pattern, and it is available to anyone who understands it clearly enough to begin.